So we're going into chapter 6, talk a little bit about genetics. This one's a little tricky. It says, son, I hear you failed genetics. But of course, if you know any families, you know that not just because everybody's got dark hair and there's one kid with blonde hair, that there's not something dicey going on genetically. It just turns out that hair color is not a single code trait. So you get variation in the family because it's a mixture of different codes. And everybody in the family has a mixture of different codes. And sometimes you wind up with light, sometimes you wind up with dark. There's a number of features like that. Now we're not going to get too far in to the weeds on genetics, but there is a bunch of stuff that I need to cover in order for other stuff to make sense. I'm going to tell you a story that I normally would tell in the lab, but the people that have to do this remotely, I didn't want to make that video too long, and I told them it'll be in the lecture video. Here's the lecture video. It's about this guy, Mendel. Well, I guess that's as good a focus as I'm going to get. Gregor Mendel, who lived in what's now Czechia, or Czechia, was part of the Austro-Hungarian Empire at that point, and got support from his monastery to go and do studies at the university in Vienna. Learned a lot about science and math from 150 years ago. Came back to the monastery and became a monk, and was tasked with, it was like a lot of monasteries. Monasteries don't tend to get by on contributions. They tend to do stuff and sell stuff, right? A lot of the surviving ones produce wine. This one happened to be a farming monastery, and he was tasked with raising peas to sell. So he was a monk who became a pea farmer, and eventually did a bunch of studies about where things come from, where offspring traits come from compared to the parents. Must have taken him a while of growing peas to notice that there are some features in peas that come in either or varieties. Not a whole range. You get purple flowers, you get white flowers. You don't get a whole bunch of light purple flowers in between. For his particular group, he got tall plants and short plants. He was good at growing them, so he grew them well, and they were tall, and no matter what he did, some of them were short. And some of the peas were distinctly yellow, some of them were deep green. Some of the pods were distinctly yellow shade, and some of them were green. But a yellow pod could have green peas in it, or green pea, or yellow peas. So he found a bunch of these either/or traits, and he figured, okay, this is simple. What's going on here? What's making this happen? And he must have subdivided whatever land he had, and he did some research, background research, which a good scientist does. Now, pea flowers, like most flowers, are sexually reproductive structures. We'll talk about that a little bit later. And they have male parts and female parts. The male parts produce the pollen, the female parts are down in the base of the flower. They produce the egg cells, and they generate the seeds, or in this case, the peas. And he found out that you could control parentage in pea plants by going to the flowers individually, before they've opened up, opening them gently, taking the male structures down, which is they're in a little ring inside the flower, taking those out, putting them in a little paper envelope, marking the envelope what flower it came from, and then taking cheesecloth and wrapping the flower so the pollen from the outside world could not get into that flower. And then you leave them. Eventually the flower opens up, it changes color. That means that the female parts, the egg cells, are mature. The male parts have matured inside the envelope. They're now generating pollen. Pollen is a sperm casing. Again, we'll talk about that later. And then you can go back to whatever flower you've selected and take pollen from the starting flower and douse it on the female part of that flower. Now when you get peas, you know who the mommy flower and the daddy flower were. I can't imagine doing this on a field full of pea plants, but this is what he did. What he eventually got to, because this is what he needed just to start his experiments, he got a plot where as long as he reproduced within the plot, he would always get tall plants, and he got a plot where he always got short plants. So one where he always got purple flowers, one where he always got white flowers, one where he got yellow peas and green peas, and there were a lot of traits. Because his question was, these things are just crossing each other randomly out in the field. What happens when I take a purple flower and cross it with a white flower where I absolutely positively know that's what happened? What happens if I cross where the male is this, the female is this, the female is this, the male is this? It turns out that that didn't matter, but he had to do that. But he needed plots around that were what we would call pure breeding plots that only had one of the paired traits on. And then he needed more plots to plant the peas from the crossings. When he crossed the purple and the white, and he planted those peas, all he got was purple. When he crossed the yellow peas with the green peas, all he got was yellow. And that kept happening, is that he would cross the two either or traits, plant those peas, and only one trait would show up. Now he's a smart guy. If every time these things crossed in nature, the one that didn't show up was just gone, was just obliterated, then it wouldn't be very long, not too many generations before it would just be gone. So he goes, okay, something else is going on here. How do I figure out if it's in there? How do I see that it's in there? Well, why don't I take the peas that I know are a cross between purple and white and cross them. Purple-white hybrids, yellow-green hybrids, a whole bunch of yellow-green hybrids. And then, tall, short. I need more plots, so he had a plot for his hybrids. When he crossed the purple-white hybrids and he planted those peas, he got white flowers, the flowers that had disappeared in the first generation. He got them about a quarter of the time. And that kept happening over and over and over. The hidden trait would reappear a quarter of the time. And from that, he figured out, for whatever trait you're looking at, there's a gene that makes that trait happen. And all of these peas have 2 copies of that gene. But the codes may not be for the same variation on that trait. There's a trait for flower color. You can have an allele, a variation, for purple, and an allele for white. When you put the 2 of them together, the purple hides that the white is there. But when you cross hybrids, the combination of white paired up with white again happens about a quarter of the time. And from that told him that he was dealing with pairs. And he came up with the dominant and recessive stuff. And he came up with all sorts of ways to write it down, which we'll deal with in the lab, and how the crosses work. And I'll show you in the lab where that quarter of the time comes from. And keep in mind, peas have a lot of traits. They're not all dominant recessive, but this was a simple way in to the basic ideas of genetics. So he wrote up a bunch of papers and he went, well, what if I do this and do the tall short at the same time? And 2 dominants didn't affect one another, 2 recessives didn't affect one another. He decided that all the genes were somehow separate from each other inside the peas. Turns out he was wrong. And we'll talk about that in the lab. But he sent all these papers out. He published all these papers. This stuff is very math heavy. Modern biologists, not that crazy about math. Biologists 150 years ago, even less crazy about math. And nobody seemed to be reading his papers. How do you know? Because people write papers and use you as a reference. They build on the work that you're doing and they use you as a reference, and that can be tracked. It's what they call a citation index. Nobody was doing that. Is that historians believe that they were reading the papers, but the math was too daunting for them, so they didn't feel comfortable going on with it. And Mendel eventually, after doing many papers, just kind of said, you know, the heck with this. I'll run the monastery, which he was doing at that point. And his papers just kind of sat there until the world of biologists became a little bit more math savvy. Remember postmodernism? And math savvy biologists started to dig through what had already been written about this stuff, and they found Mendel's papers, went, oh great, here's the rules, here's how we can write this stuff down, and he's now considered to be the granddaddy of genetics. So with that as kind of the basis, and it's kind of the lead into the lab for the genetics lab. We'll talk about some of the terms. For Mendel, a gene generated a trait. No accident, the same words in there. We now know a lot more about genes. And it isn't a trait that's produced by the gene, it is a protein. For the flower color, the gene that Mendel was working with is a gene for purple pigment. An allele is the DNA code, and it's a variation. Now, there are many different alleles for the same gene. DNA codes are very long. You can get little changes in them. Most of the time, the protein that's being coded for does exactly the same thing it was supposed to do. Sometimes it does something different. Sometimes it does something better. If you're going to change what it does, most of the time it's going to do something worse, right? You're going to take something that's working, you're more likely to mess it up than to help it. Ask anybody who's experimented with recipes where they just kind of change things randomly, because that's what alleles are. Alleles are random mutations. We'll talk about that in a minute. Now, here's what happens with dominant recessive. Sometimes you get a code and it makes a version of the protein that doesn't work. It doesn't do anything. In the case of the purple pigment, the recessive allele didn't produce a pigment. Now, the base color of the flowers is white. That made the flowers with just two recessives, two bad codes basically, white. Turns out the height gene, Mendel, was a gene for growth hormone. His short plants had two codes that produced the growth hormone that didn't work. Now, it turns out you only need one working code for growth hormone to get a normal height plant. You only need one code for purple in order to get a purple flower. So a presence of dominant and recessive, the dominant hides that the recessive is there. Working covers up non-working, right? I'm sure you've all been in jobs where, you know, one person's doing all the work and it kind of covers for the group that, you know, some people not pulling their weight. But this is how dominant recessive works. Most of the time, alleles are not this all or nothing product in the proteins, and sometimes you get stuff and it mixes together. We'll cover that when we get to the lab. So recessive is generally a non-working protein or very weakly working protein. And This is almost always when there are genetic diseases, this is where it comes from. And genetic diseases are almost always recessive. Sickle cell anemia. Sickle cell anemia, you have an oxygen-carrying protein in your blood, your nice round red blood cells. This oxygen-carrying protein, hemoglobin, is a nice round protein. If you get the sickle cell allele, the protein that you make is round when there's oxygen stuck on it, but when oxygen comes off of it, it changes shape. It gets kind of long and skinny and curved a bit. And in a red blood cell, if that's the only hemoglobin you've got, you have two recessive codes, and that produces this defective hemoglobin. Anytime your red blood cells get a little bit oxygen depleted, enough of those proteins have changed shape that it changes the whole shape of the cell. It sickles them. It makes them little C shapes, and they don't go through little blood vessels real well. Sickle cell anemia is pretty dangerous. It can kill you. Now, here's a situation where if you have one sickle cell allele and one regular allele, then half of the hemoglobin you're making is sickle cell, is the sickle protein. But your cells have to get really oxygen depleted for there to be enough of them sickled to change the overall shape of the cell. So most people with sickle cell trait don't ever experience issues. Playing football in Denver, sometimes high altitude stuff can mess with them, but they really need to be oxygen depleted. And there's other things. Cystic fibrosis is that there is a protein that adjusts the wateriness of the mucus in your systems, your breathing system and your digestive system that way, of your mucus. And if you have the recessive allele, that protein doesn't do it effectively, and your mucus is thicker than it's supposed to be, and it doesn't move the way that it's supposed to. It jams up. It can clog your breathing tubes. It can clog your digestive system. It can be pretty dangerous. Now, one of the questions about genetic diseases has always been, okay, evolution should have gotten rid of those, right? Is that anybody who had them through a mutation, they would have had issues that should have been removed from the population. Why are these things found? And in some populations, they're not real common, but they're way more common than you would think they would be evolutionarily. Eventually, they discovered something interesting about a lot of these diseases. You have a good normal allele making the protein it's supposed to make, and a bad recessive allele making a non-working protein. If you carry two of the good ones or two of the bad ones, pretty easy to tell what two of the bad ones are doing to you. You know what happens if you have one sickle cell allele and one regular hemoglobin allele? I just mentioned, if your cells get really oxygen depleted, it can mess with them. People that are hybrids, we'll talk about this later. Malaria reproduces inside red blood cells in our little bags of hemoglobin. They get in there and they make a lot of little malarial parasites. That depletes the oxygen in the cell crinkles up. Your body goes, well, that's worn out. Let's tear it apart and recycle everything. Kills the malaria doing that. So if you have one sickle cell allele and one regular allele and you're exposed to malaria, you have a much better chance of surviving it because a lot of the malaria is eliminated through this weeding out process. In areas where there's malaria, two sickle cell alleles, pretty bad. You get sickle cell, that's bad. But two good alleles, you're more likely to die of malaria than the hybrids. So that has made sickle cell allele valuable evolutionarily in that population. It's become more common. Cystic fibrosis turns out to produce a resistance to tuberculosis, which messes with the mucus in your systems. Is that schizophrenia may be the wrong mix. It's not a single gene, it's a whole bunch of genes, but it may be a single bad mix of genes that are very behaviorally useful in groups. So hybrid vigor is just a feature that people were very surprised to find. We'll get one more thing out of the way. We are making copies of our DNA and moving it around all the time. I'm making new skin cells under here. Every new skin cell's got to get all of the codes it needs in order to be an active working skin cell. Now, we have thousands and thousands of different genes. If we needed every time we made a cell to make copies of all of those separately and then move them one whole set into one cell, one whole set into another cell, we would mess that up all the time. We need our DNA to be in a number of pieces where we can copy it, we can move it, we can distribute it, and we're not going to mess it up that often. That's chromosomes. One chromosome has thousands of genes on it. Not even doing the same basic sort of stuff, except for the Y chromosome. But all your others are just this mishmash of different genes doing different things. It's just a matter of having a trackable number of pieces. We have 46 chromosomes. Every time we make new skin cells, we make copies of each one of those. Now we got 92. We're going to get 46 to go into one new cell, 46 to go into the other new cell. It's got to be the right 46 so that the cells have what they need. And so chromosomes reduce the number of DNA pieces to a manageable number. Genes that are on the same chromosome are considered to be linked. I'm squeezing this in here. You're going to want this on separate lines. Pretty soon they found out Mendel was wrong, that all the genes are not completely separate from one another. It took a long time to figure out that some of them were riding on chromosomes together, so they get passed on as packages. They're connected to each other. So, you know, if cystic fibrosis and sickle cell were genes on the same chromosome, then if you had a sickle cell allele on the chromosome and a cystic fibrosis allele on the chromosome, they would go to your offspring together, even though they're not doing the same thing. Chromosome numbers. Different species have different chromosome numbers. We have 46, chimps have 48. C. elegans, which is a worm we'll talk about later, has 4. And it's a characteristic of every species. And some of them have a lot of numbers. 46 is a fairly high number for an animal. Some of them have very low numbers. 4 is a pretty low number for an animal. There's upsides and downsides for these. If you have a low number, that's just a few pieces of chromosomes to keep track of. So when you make new cells, when cells divide and you distribute the chromosomes, you're not going to mess it up very often. So you don't get a lot of division mistakes with a low number. But we have 46 chromosomes. We have 23 pairs. And when we make offspring, we put 23 chromosomes into a sperm cell or an egg cell. And the way we can mix those is, there's a lot of different mixes you can get when you've got 23 of each to select from. Remember from the evolution chapter is that variation is a big deal in evolution. Variation within a population is, there's survivors and, not survivors because of variation. So high number gives you more variation, but also more mistakes when you make cells divide. Low gives you low variation, but fewer mistakes when cells divide. And that's the trade-off. You see this a lot. The advantage of one turns out to be the disadvantage of another. That's going to turn out to be true for what we're going to cover in the next video.